Okay, good evening. We shall start now our running course BTL 611 Probiotics, Prebiotics, and Postbiotics. In our previous lecture, we started a talk on plant probiotics. Today, we shall continue a bit and then we shall move to 31st celebration lecture and a gathering. We discussed about the discovery of plant probiotic bacteria. In our laboratory, we have been working on this topic since 2010 to discover plant probiotic bacteria for using them as biofertilizer or biopesticide or biostimulant. And our strategy is unique because I received considerable training on ecological chemistry when I have been in Japan. In ecological chemistry, the basic principle is every organism depend on others and they are interconnected and interdependent. We targeted probiotic bacteria from plant, mainly the endophytes, not the saprophyte and other organisms available in the environment and test them. So our approach is a bit different. Major sources we use to collect or isolate the bacteria: plant seeds, stem, root, and rhizosphere. Uh, Bangladesh is the largest delta in the world, and rice cultivation is one of the primitive agricultural practices in this land. And rice and fish is our life. Rice is like our life. So we had lots of local varieties adapted to this delta. And this is why we thought it is better to target bacteria from the locally adapted varieties. So we collected a large number of locally adapted varieties from an NGO, Unnoyan Dhara. And then our mission was to surface sterilize the seed and from the internal tissue uh, to discover the bacteria. And they are specific because they are co-evolved with rice in the ecosystem of Bangladesh. Uh, and our expectation is that if we do this job and if we find some interesting bacteria, endophytic bacteria, uh, those bacteria could be used as biofertilizer because they can easily colonize the rice plant. And in these are the pure bacterial, bacteria uh, purification process is a bit lengthy. First, by using dilution technique, you get the uh, crush the plant seed in uh, sterilized water and then dilute it, for example, 1 million times or 10,000 or 100,000 times, and then spread the liquid on the agar medium so that you can get the distinct colony next day or within a few days. And then by using toothpick, you can pick up the unique colony which you think almost pure. And then by this strict culture, we can spread on the medium and then we see whether homogeneously the color and colony is grown or not. If it is homogeneously grown, then it is pure. Otherwise, second time, again dilution technique you have to do to separate the bacteria. And one of the simplest bioassay we use in case of rice probiotics uh, to germinate the rice seeds in the petri dish uh, in sterilized water without any nutrition in presence and absence of bacteria. Here you can see this is the control. Control germinated with uh, epicotyle and hypocotyle, root part and shoot part. But you see normal germination, not so branching, and this is common in case of rice. But here, uh, BTS5, one of the strain of bacteria, or BTS1 or BTS6, the growth of root and shoot considerably higher than the control. And we allowed them to grow 14 days on the petri dish without offering any nutrition. And we found that uh, after 14 days, the dry matter in some bacteria treated one increased 3 to 4 fold than the control. Now the question is how that dry matter increased. One way is fixation of nitrogen from the atmosphere by the bacteria, and also phytohormone production, and other process of triggering the gene expression in the host so that they can make more dry matter through a photosynthesis process. So this is really amazing, and this small bioassay technique can give you a big canvas for doing fundamental research to understand how tiny bacteria interact with rice root and rice plant and enhance the growth. We tested them not only in the petri dish, we tested them in the pot experiment. And interestingly, we applied different doses of fertilizer with bacteria and without bacteria. We cut 50% fertilizer in some pots, and some pot we used full doses of fertilizer. But interestingly, when we cut 50% of fertilizer and use the bacteria like Serratia marcescens, we found that the growth, root and shoot growth, even 50% reduced fertilizer are equivalent to the full doses of NPK fertilizer used in the pot. And that means bacteria can cover 50% of synthetic supplement 50% of synthetic NPK fertilizer. Now, question is how? Nitrogen, it is understandable that nitrogen can be fixed by the bacteria from the atmosphere and offered to the plant. But in case of phosphorus and potash, these two elements, they are uh, present in the soil in fixed form, that means insoluble form, organic or inorganic. Bacteria, through the enzymatic degradation and mineralization process, they solubilize the P and K and offer to the plant for nutrition. This is why we can reduce fertilizer NPK about 50% and get the same yield of the full recommended doses of fertilizers. We did experiment in the pot and also check the grain yield. You see, full doses of fertilizer when we used per pot, we got the this mass of grain yield. But when we used half doses of fertilizer, you see, we found some of the bacterium BTS2 gave the higher yield than the full doses of fertilizer, although 50% fertilizer is reduced. But how about without fertilizer? Without fertilizer, actually, there is a yield penalty. Bacteria cannot compensate and give the result of full doses of fertilizer. That means bacteria has some limitation. Certain amount of fertilizer can be supplemented or provided by the application of bacteria. One of the greatest benefit of using bacteria instead of chemical fertilizer is that bacteria, they are living organism. When you apply in the seed as seed coating or seedling root dipping into the bacterial suspension, they colonize and form biofilm. And when plant grow, bacteria also grow and their number also increase. But in case of fertilizer, from the application uh, and through the time, uh, they degrade, they are lost through various ways, volatile and leaching, and also plant uptake. So gradually decrease, and at the certain period of time, you cannot find any residue. But in case of bacteria, they grow with the plant growth. Other point is that bacteria, after their death, they add organic matter, and they also, you know, modulate the microbiome in the rhizosphere as well as in the plant body. Microbiome means positive microbes. The number of positive microbes proportion are increased. So there are lots of benefit there. So here you can see this is the control. uh, this is half dose, this is full dose. You see many bacteria with half dose, they gave the higher grain yield than the, the control. Control means full doses of fertilizer. And our result confirmed that 50% NPK fertilizer can be saved. And it is, about billion taka business in Bangladesh. So we checked after, seed treatment as well as root colonization when plant grow, whether bacteria also colonize on the rhizosphere or not. By using scanning electron microscope, you can see all dots are bacteria. So the bacteria you applied, they dominate in the rhizosphere, and you modulated the population, and they exert the beneficial effect, which is easily visible. In our BSMREO field, we did an experiment. Here you see BN8, one of the bacteria, uh, gave the higher yield than the half and full doses of fertilizer with half doses of fertilizer, and you cannot distinguish them with the recommended full doses of fertilizer. Very amazing. And we also tested them in the field, and we also tested the same behavior or function in the wheat. That means they can be used in wheat also, other crop also, and in the strawberry, we found very profound activity. 43% yield of strawberry uh, was increased by the application of some probiotic bacteria. So probiotic bacteria not only increase the yield, we found other thing. We found that probiotic bacteria increase the yield as well as antioxidant contents in the strawberry fruit. And that paper was published in scientific reports. Here you see antioxidant like total flavonoid content, total phenolic content, or total antioxidant activity, total carotenoid content, total anthocyanin content. This is control, and these are the two bacteria, Parabarcolderia and Bacillus. Amazingly, they are increased by the application of the probiotic bacteria, and it is the first report that you can improve the quality of the agricultural produce by the application of the bacteria. So this is all about today's lecture. And in our lab, we have a big bank of 650 bacteria we discovered from different plants, and some of them are identified through 16S rRNA gene sequencing, and some of them we did, a whole genome sequencing, and some of them we found very powerful biopesticide and mechanism of their growth promotion as well as disease control we are now studying, and some of the mechanisms are known. So next class we shall discuss in detail. So this is all about today's talk, and today is the last day of the year 2020. 2020 really in the human history, it would be regarded as a uh, remarkable uh, pandemic year, COVID-19. And today I heard 15,000 people died in a day. That means killed nearly 2 million people worldwide. COVID-19 is posing still a serious threat to the human life, human other activity. We are now attending class online instead of face-to-face interaction. Uh, our university dormitories and laboratories are almost, vacant. This is really a horrible situation. We hope 2021 would be the recovering year, and that would be the uh, much more better than this, horrible year. But anyway, what is most important, uh, the philosophical point is to adapt to the any adverse situation, uh, not only for human, for all organisms. So we are adapting, and a new normal, like online meeting is now new normal. Every year we organize 31st to celebrate the year, what we achieved, as well as I always, since 2010, offer a seminar on the Nobel winning topic of that year and try to relate with our own research so that we can cheer up us for the new year to do something outstanding. Thank you so much. So if you have any question, you have still 5 minutes time. You can ask question. So do anyone have any question?
No, sir.
Oh, thank you. And how
No,
about
sir.
Ruhul Amin?
Oh, wishing you sound health in 2021.
Same to you, both of you, and all of our, IBG family members and all the humans in this earth. We should pray for their sound health as well as prosperous life. And obviously, uh, we should wish to win the fight against the tiny coronavirus and get a new momentum. Thank you so much. So, 8 o'clock, please join to celebrate the just after 4 minutes, the 31st, uh, in a virtual way. Thank you so much.